Good morning, everyone. I'm glad to see that some of us have made it here. Sounds like it's a bit of chaos with people not being able to get here with flights and people being unwell, so it is great that some of us have made it here on time and we can kick off the conference with the educational courses. So unfortunately, my co-chair Anna has been subject to some travel issues as well, so she's currently stranded in London, so we're hoping she can join us later on today. So we're here for this session towards translation, improving preclinical stroke pipeline, and we've got a lineup of fantastic speakers for you. We're going to kick off with Dr. Emily Senna, who's a senior lecturer at the University of Edinburgh, and she's going to be speaking to us about preclinical multi-centre studies, why, how and when. And I'm sure you'll agree Emily is really well versed to speak to us on this topic. So great to see Emily, and I'll hand over the floor to you. Thank you so much.
Thanks, Renee. I'm very impressed that we have an audience at this time of the morning on a Sunday. So thank you all for making it here. As Renee said, my name's Emily Senna, and I'm based not too far from here at the University of Edinburgh. I always start with my disclosures, so you know, I guess, my biases, because I talk a lot about bias in preclinical research, so you know what my perspective is. I'll just let you quickly have a look at those. And I think it's also helpful for me to give you my perspective in terms of where I'm coming from in regard to the research that I do. So I describe myself as a preclinical meta-research scientist, albeit with a focus in stroke. Essentially, I'm interested in the modelling of human diseases in preclinical models in the laboratory, but my focus really is on trying to, I guess, shine a lens on how we design, conduct, report preclinical studies to try and improve them to increase the likelihood of translation. So this is a slide that, and I really wanted not to start with this slide, but because it's such an old slide, and if you've ever seen me speak before, you'd have seen these data, but I think it really characterises the problem that we have in preclinical research in terms of translational failure. This review was done by colleagues in Australia back in 2006 now, in which they identified over 1,000 different interventions that have been tested in the model of stroke, 600 in animal models, 374 have been have been shown to be effective in these animal models. Just under 100 taken forward to be tested in a clinical trial, and only one intervention, clot busting with thrombolysis, was shown to be effective in these trials. I guess now we do have, and we do have other interventions that are effective clinically, but they don't have the supporting animal data. And to be honest, I think showing the data like this is slightly disingenuous, because it's not that TPA was shown to be effective in these different steps before it was taken forward to be tested in humans, but it was data from myocardial infarction studies and things that led to where we are now. So thinking about translational failure, our approach has been trying to understand what are the potential sources of bias that might be contributing to these issues. And there's kind of 4 areas that we've focused our research on. The first is the internal validity of a study. So that's the strength of the cause-effect relationship that we've seen in an experiment. Are the effects that we observe because of the intervention that we've given, or are they due to other unknown systematic biases? And things that you can do to strengthen the internal validity of an experiment are random allocation, blinding, using appropriate controls, that kind of thing. External validity, that is the extent in which you can generalize your findings to different settings. Obviously, when we do an experiment in a lab, it's not just to show an effect in a lab. We want those findings to generalize to other settings, to other people, to replicate, to reproduce those findings, but ultimately for those findings to be applicable and to translate to humans in a clinical setting. Construct validity, so this is really the concept of, I guess, understanding understanding, are we measuring what we think we're measuring? So do the models capture what we think they're capturing in the disease? Are the outcome measures that we're using appropriate to then translate to different settings? And then finally, publication bias. This is the phenomena that neutral and negative studies generally either take longer to get published or don't get published for all sorts of different reasons. Primarily, I think, because of the incentive structures that we have in academia that don't reward and incentivize us to get those types of data out, but also, it's just not as interesting. When you get a good, exciting finding, that's what engages you, that's what you want to write the paper about, that's what you want to get up and tell people about. But I think, it's important that all the knowledge that we generate is distilled and shared, and that helps us move things forward. So we've looked not just in stroke, but in the modelling of lots of different diseases at some of the prevalence and impact of these biases that I've described. We've looked at stroke, motor neuron disease, Alzheimer's, Parkinson's, EAE, model of MS, and glioma, and we see very few studies report things like randomisation or blinding the assessment of outcome. And when we look at the impact of this, it appears to have an effect on the effect sizes that we observe. So these are data from a systematic review that we did a few years ago, where overall the intervention in question appears to improve outcome by over 40%, a very effective drug. But then when we stratify the data according to studies that report randomization versus those studies that don't report randomization, we see much larger overstatements of treatment effects in studies that aren't reporting such measures to reduce the risk of bias. And this is just one example, but we see this consistently across different settings. And like many of the other interventions, the intervention in question here, when it was tested in the clinic, didn't work. I feel like for years I was always kind of the naysayer saying these are all the things we do wrong and look at the impact of all these things that don't go well. But I do want to say that things are improving in terms of internal validity or the reporting of measures to reduce risks of bias. So back in 2009, we did a systematic review looking at the effect of interleukin-1 receptor antagonists in models of stroke and showed very limited reporting of randomization, blinding. None of the studies that we looked at reported a sample size calculation. And there's kind of, and we can talk about it in the break or in the Q&A, there was kind of a whole host of discussions that happened afterwards about, people who were involved in IO1RA studies saying, well, just because we didn't report it doesn't mean we didn't do it. But my response was, well, if you don't tell us you did it, then no one will know that you did it. So despite the kind of pushback to our initial review, there was this reflection that actually, if people are going to be able to use, if we want people to be able to use our data, then making sure that we fully report what we've done is important. And I know Matt's going to talk about reporting, I think, in the next talk. But it appears our review did have an impact. And when we went back in 2016 and did another update of the interleukin-1 receptor antagonist literature, we saw a huge increase in the reporting of measures to reduce the risk of bias. So I feel like we are, particularly in the stroke field, actually compared to other disciplines that I've looked at, or other domains that I've looked at, we are going in this positive direction, which is really encouraging. Moving on to external validity, so this is the generalizability of findings from one lab to, like I say, other labs, but also to other species, including humans. I think one of the things that has limited this is what we've described as the standardization fallacy. And essentially, this is in our efforts to increase reproducibility, we try to reduce variation by standardizing everything, because we think if we standardize everything, then the next person, if they follow those standards, they'll be able to get the same result. So we do that in the lab environment, the tests that we use, the highly inbred strains of animals that we use. But actually, by that increasing that standardization, what we're doing is increasing the risk of detecting an effect with very low external validity. So that effect will only be observed in those very standard conditions, and those standard conditions aren't going to be relevant to a human who has a stroke in different settings. And we've tried to describe this using this description called the reaction norm. So if we think that the effects that we observe are an interaction of a phenotype of the subject and its environment, then depending on some sort of nuisance variable that might be happening in that environment, you might observe an effect on one area of the action norm, and that could just be the radio's on in the lab, or if, I don't know, I'm thinking kind of random here, but if, I don't know, I come in wearing a different perfume one day, you might observe a effect on a different area of the reaction norm. And then we characterize this as a failure to reproduce. But I don't think it is a failure to reproduce. I think what it is that we're selecting different areas of the reaction norm to see our effect. And we've done some modelling work, to try and, increase reproducibility by introducing standardized heterogeneity and reducing that standardization using a multi-center approach. And essentially what we've shown is, and these are simulated studies, the dotted line here is the pooled effect, it's the true effect of all these different individual experiments that are done for the same intervention. And what we see is that where you have one lab doing an experiment, you get sampling different areas, and it looks like we failed to reproduce. This study, sorry, the mouse is a bit glitchy, so I'm not getting great. You get, some studies that don't cross, this is a 95% interval of the effect size. There's no overlap with that 95% confidence interval, so it looks like there's no reproducibility. When you do multi-center studies, and then you're putting in that heterogeneity into your experimental design, we see much tighter confidence intervals, and all the studies overlap with the true effect, suggesting that multi-center studies are a method to, because of that introduced heterogeneity, to increase reproducibility. That was an animation there. Construct validity. So we've not done a huge amount of work in this area, and it's where there's a lot of activity at the moment. But like I said before, this is, the concept of, are we measuring and modelling what we think we are? Models are models. They capture aspects of disease. Stroke is a clinical condition. We model focal cerebral ischemia in an animal. There's lots of different models that capture different aspects of the human condition. A multi-center study actually can be useful because it's very difficult for one lab to do lots of different models, but pooling those resources together and capturing those different aspects of a disease and showing intervention efficacy across different types of models can be useful. The outcome measures that we generally use in preclinical research are surrogates. We look at infarct size, whereas clinical trials' primary outcomes may more likely be functional, daily activities. Can someone get out of bed? Can they eat their breakfast? Can they tie their shoelaces? That kind of activity. Another thing that I think is important is time. We all know that in stroke, time is important. Time is brain. And in some of the systematic reviews that we've done in the past, we've seen a big difference in time to treatment between the animal studies and the clinical trials. Tirilazad, an antioxidant, large phase 3 clinical trial, failed to improve outcome. In the animal studies, time to treatment was 10 minutes on the median time to treatment across all the studies. TPA, thrombolysis, an effective intervention. In the animal studies, median time to treatment was 90 minutes. And then when we compare this in the clinical trials, Tirilazad, 75% of the patients were treated after 3 hours. So we're not designing the trials to reflect where we're seeing effect size and efficacy in the animal studies, whereas the TPA clinical trials were in line with what was observed in the preclinical studies. And then the final type of bias that I wanted to mention, publication bias. So like I said before, this is the phenomena that neutral and negative studies either don't get published or are less likely to get published, and then essentially what's in the public domain is systematically different to all the experiments that have been done. And we looked at trying to quantify publication bias in the preclinical stroke literature. And essentially what we did is we used a technique called a funnel plot, in which on the x-axis you plot the effect size of each of your experiments, and then on the y-axis you plot the precision. The precision is the inverse of the standard error, so the more precise your study is, the narrower the confidence intervals are. And the black dots are the experiments that we had in our data set. And what you expect when there's no publication bias is the more precise your study is, it should lie around the true effect. And then as you become less precise, i.e. when your confidence intervals start to get larger, you should have an equal distribution of positive and negative studies. What we found is that there was a skew, there was a lack of imprecise negative studies. We use a technique called trim and fill that essentially imputes these theoretically missing studies so that you get more symmetry in your funnel plot, which are these red dots here. And what we found was overall our effects reduced from 32% improvement in outcome down to 26%. And we estimated about 1 in 6 experiments remain unpublished. Now, I think this is a gross underestimation of the magnitude of this problem, because a lot of the experiments that we observed on this side of the plot, the negative studies, weren't truly negative publications. It was studies that had positive findings and negative experiments in one paper. Only 2% of our papers in our data set truly had no statistically significant positive findings in them. I've not talking a lot, I mentioned briefly sample size and lack of sample size calculations and power, but I think sample size also conspires with publication bias, because I think when you are underpowered or of unknown power and you have a positive finding, that study will get published because it's positive, irrespective of the power. Whereas when you are underpowered or unknown power and you are sitting on this side of the graph, you're less likely, that paper is less likely to be in the public domain, less likely to be published. So then this exacerbates that problem. So my title was why, when, how, I think. So the why, we should be thinking about multi-centre studies. I think there's lots of evidence that translation's failing. So just, I think, taking a different approach, looking at how we do things differently, I think is useful. Hopefully I've convinced you that reports of animal experiments seldom report measures to reduce risks of bias, although I think that is definitely improving. Experiments that are at risk of bias tend to give overstatements of treatment effects. Many studies don't generalize, and I didn't have time to put in, we talk a lot about translation, but even replication, when people try to do what other labs have done, there are huge difficulties and struggles in that space. Most studies are underpowered or of unknown power. And when we've done some secondary analyses, we see substantial publication bias and selective outcome reporting. So thinking about multi-centre studies and clinical trials versus in vivo studies, I think there's differences that we could potentially reduce. I think we could, fields can learn from each other, that I think could increase the utility, particularly of preclinical studies. So things like the number of centres, think about time response, thinking about power, the outcome measures that we're using, publication bias. It's clinical trials, you have to register a study if you want to publish it. So that, there's cultural norms that aren't cultural norms in the preclinical space that I think we could learn from. Data sharing, making data available. I think that probably isn't great in either discipline. But also regulatory and ethical processes to make it easier to do these types of studies. So ideally, I think what I'd like to see is studies would benefit from strategies that facilitate robust design so that we can increase the internal validity of an experiment, but also are able to assess its robustness. that facilitate collaborative studies so that we can increase the external validity so that then our findings are generalisable to other settings. I think clarity of how studies were performed, again, so that we can assess robustness, but also allows us to replicate where appropriate. Confirmation that studies report what they set out to do. I haven't really spoken about outcome switching and statistical analysis switching, but when you design a study, reporting what you set out to do rather than reporting only the outcomes that gave you a significant finding, there's clearly an important impact there. And then access to the data. People have got lots of data in servers and hard drives and in the cloud now, I guess, that we aren't able to use and can't learn from. A lot of the work that I do is systematic reviews, where we look at data from old experiments, and that has informed a lot of what I'm talking about today. It allows us to assess robustness again and to replicate those findings. So I'm going to talk through each of those headings and some of the tools, and I guess this is the kind of how I think we should be thinking about some of this stuff. Some of the tools that are available to assist with those 5 different headings. So for robust design, I think there's lots of great tools out there. I'm assuming Matt's going to talk about the Experimental Design Assistant, so I won't go into that. But it is a tool that I highly recommend people have a look at. Statistical input. Statisticians often gripe that people come to them when they finish the experiment and ask them how they analyse it, and essentially at that stage all they can do is give you a post-mortem of how you did things wrong. Getting stats advice at the beginning of a study to inform the design will ensure then that the analysis is appropriate. So if you have access to statisticians, and I know biomedical statisticians are like gold dust, few and far between, so be nice to them if you find one who will work with you, because I think they're definitely worth their weight in gold. But also peer review. And I'm talking about peer review before you actually start doing an experiment. I think the system that we use for peer review is happens at a stage, again, where you can get a post-mortem rather than where you can get input and influence into the design. And I'm going to speak a bit about this using a publication model called Registered Reports, essentially where you design your idea, design your study, it then goes for peer review at that stage. This also has an impact on publication bias because you get in-principle acceptance on your study, you get input on your study at the design stage. It then changes the incentives because then it's not about getting the right answer in terms of effects, but it's about following what you said you're going to do and following the robust design. You then collect the data, write the report, you go for your second stage. Your second stage is really about making sure you did what you said you were going to do, rather than the spin, and then publish your report. So peer review before data collection. There's a platform called Peer Community in Registered Reports that I'm, full disclosure, one of the managing founders of. And essentially this is a journal agnostic platform. So it's essentially what we do is we peer review preprints. So before they go to a journal, you go through the process at that preprint stage, and then once the study's finished, you can then submit it to any journal that will take it. But you've got that robust history and, I guess, audit trail of saying what you plan to do, having that peer review input, doing the design, and then having the report at the end. Like I say, I think it emphasises the importance of the research question itself and the quality of the methods, and it's designed to eliminate things like low statistical power, selective reporting of results, publication bias. But it does allow for complete flexibility to report serendipitous findings. People get really kind of stressed, but I didn't think when I wrote my plan that I would find this thing and I want to write about this thing, and you can write about that thing. You just say this wasn't a pre-planned analysis, it was a post hoc thing, and people can then interpret that as they see appropriate. Collaborative studies, like I said, to improve external validity. I'm going to speak a little bit in a minute about multi-part. So this was a project that I was involved in a few years ago, where we sought to try and I guess think about the different aspects of a multi-centre study. thinking about systemic heterogeneity, improving the constructs that we do. One of the big lessons from this is don't try and do it on the cheap. And as with all academic research, getting funding to do things is difficult, but it's important that you do get the appropriate resource, because this is an added level of work. And I haven't also spoken much about incentive structures in academia, but I think this is, and it came up in the multi-part discussions, well, who's going to be first and last if you've got all these labs contributing to a study? And I think we need to start shifting how we think about how we credit authors and participants in research. I'm a big fan of ORCID. If there's one take home from today, get an ORCID ID. But the credit taxonomy is great, and quite a few journals now use the credit taxonomy. So personally, I would love it if we used, we had like film credits for, so you really know what people did. And if I want to find somebody in a paper who did, I don't know, the statistical analyses or the Western blots, whatever, I know who that person is, rather than just saying, the first and last, and actually having no detail. And the credit tries to kind of start to build on that, who designed the study, who actually carried out the study, who did the analysis, who did what sections. So I encourage people to engage with that as well. Clarity on what was done. There's some great tools out there, things like protocols.io, which allow you to, essentially, it's an online platform for your SOPs, so you can have details, especially when you're doing collaborative studies. You can share that information. If there's deviations or changes, they can be added by different collaborators. Electronic lab notebooks, I think there's been a big shift to that space in recent years. Research identification initiative IDs, RIDs, so people know what reagents, what antibodies, what specific things have been used, and then obviously reporting studies appropriately, and the ARRIVE guidelines that, again, I think Matt will talk about, so I won't go into more detail there. Confirmation that studies report what they set out to do, using trial registries, I think would be fab, especially for confirmatory studies. I don't think all studies necessarily it's appropriate. I can understand with exploratory research it maybe doesn't fit as well. But when you're at the stage of doing a multi-centre study, then I think pre-registration of a trial would work very well. There's different platforms out there, preclinicaltrials.eu, there's the Animal Study Registry, or there's domain agnostic registries like the Open Science Framework, in which you could pre-register a study. There's also been, I think, a shift in recent years in the preclinical space to publish study protocols, and of course registered reports would also get around that. And then finally, access to data. So that it can be used and shared efficiently. And I think there's kind of two levels here. There's getting data out quickly. And I think that preprints are a great resource for that. We did a study at Edinburgh where we looked at time from ethical approval of of an animal study through to publication, and 2/3 of the time was spent at journals. And I'm a journal editor, but and I understand the complexities. But I think that's outrageous. That really delays our progress. If we can get information out, there's no reason why, when it's out on a preprint, that it still doesn't go through that peer review process. But getting that information out quickly, I think, is important. And we've got great platforms that allow for that. But also making sure that the underlying data supporting studies are made available. Again, there's lots of different platforms that exist out there. I think what happened initially was that these platforms ended up kind of being homes for undocumented data dumps. People are like, my data's available. It's an Excel spreadsheet that you can have a look at, but unless you were that, and even if you were that person, 3 years later, you probably can't make head nor tail of what the different columns mean. No quality control. They weren't often linked to the original studies, and you didn't know how to reanalyze the data even if you wanted to. So there's been a big push around making data fair, findable, accessible, interoperable and reusable. And at least in the UK, but I'm sure this is probably a general thing, institutions are really putting a lot of time and effort into supporting academics to make their data fair. So if you have those resources in your institution, I highly recommend engaging with them, but also there's great resources out there that are freely available to anybody. So I quickly wanted to touch on Multipart, which was our group that was kind of planning the how we might do multi-centre studies. It was a consortium, like I say, to establish a framework for them. It was a 2-year EU-funded project. We completed, I was doing this last night, and I was like, bloody hell, 2015 was a while ago. We had 10 participants from 7 countries, and we were using Stroke as a worked example. And really it was to inform the design and conduct of adequately powered multi-centre studies with improved internal and external validity. We had 6 different work packages that we described as the different parts of the engine, and the purpose of the project was to try and build this engine together so that we had something that would run. And each work package was jointly led by an expert in that theme, so somebody who had expertise in experimental design or regulation and ethics, for example, and then an in vivo practitioner, so somebody who actually did these animal studies and could give feedback on the feasibility and the practicality of the solutions that we were proposing. So I will quickly go through, conscious I've been talking for ages now, the considerations that we came up with in Multipart for multi-centre studies. Essentially, we thought it was important to ensure that the practical aspects of organising a multi-centre study were considered. So getting accurate costings, understanding what you require from each of the study sites, how are you going to recruit those different study sites and improve them, thinking about your scientific coordination, how are you going to have your standard operating procedures, getting agreement and definition of your models and your outcomes, thinking about whether you need and who will be on things like a steering committee, a data monitoring committee, quality control committees. Having strategies to maximise the internal and external validity. So, thinking about the experimental design, making sure that your studies are powered, thinking about how are you going to have this variation across the different sites. So thinking about mixed model statistics, treating different labs as random factors, thinking about the sample of labs that you have. They should really represent the population of labs in the same way that any sample should represent the population that it's intended to. The number of labs should allow for us to have between lab variation, so we can assess that. And then the number of animals within a lab should allow for within lab variation to be assessed as well. So getting all those, thinking about all those different factors, ensuring ethical and regulatory requirements are met, obviously, ensuring a robust and secure online data management system, so that you can set up your study, that you can do things like randomise the animals, that you can capture your outcomes, monitor progress, feedback, outcomes and data for assessment and analysis, and also that you've got an appropriate and robust statistical analysis framework. In terms of when we should be doing multi-centre studies, I don't think every in vivo study should be part of a multi-centre programme. I think there's lots of hypothesis generating and testing experiments that can be effectively done in one laboratory. I think that's totally appropriate. But I think when we're in that space of trying to confirm efficacy in a robust and intensively monitored experiment with transparent analysis and reporting, then I think multi-centre studies have a place. So this is the kind of pipeline that I envisage, is that you do have these kind of single lab studies that people do. You might do a systematic review, meta-analysis, to where you pull together data from different labs, thinking about how effective a treatment is, what's the quality, is there publication bias, what are the conditions of maximum efficacy. Then that's when I think you then might start thinking about multi-centre studies, so that you can confirm that efficacy, that you can do it under these conditions that I've talked about, and then start thinking about clinical trials. So my take-homes are the in vivo studies which don't report simple measures to avoid bias give much larger treatment effects. Most don't report these things, but improvements are happening. We generally work in silos with limited generalizability, and I do think that is largely driven by our incentive structure. I don't think what's good for scientists is necessarily what's good for science, and kind of getting that balance right, I think, is something that academia is grappling with at the moment. Reporting biases are important and prevalent. So I think most studies are underpowered, or at least of unknown power. I think multi-centre studies can both directly help with some of these things, but also the nature of collaboration, I think, can also indirectly help with some of these issues. There are some useful tools that already exist, so we don't have to reinvent everything from scratch. But I do think development and implementation is going to require more resources than we have to hand at the moment. So thank you very much for listening. This is the multi-part team, and this is my research team in Edinburgh.
Thanks so much, Emily. I'll open the floor to questions, and we do have a mic in the middle of the room for the benefit of everyone who's joining us online, so welcome to everyone who's streaming. So if anyone has a question for Emily, I'll get you to use the microphone or speak really loudly.
Thank you so much for bringing up this really important point. I am Sandra Cho from USA. So you have identified a lot of issues and how we can actually move to translation. But the one thing that I was...
Sorry, could I just interrupt you? Our tech down the back is saying we can't hear you. Would you be able to go to the microphone in the middle of the room? Thank you so much. Thank you.
Okay, so one thing that I was always wondering is the preclinical studies and then the human studies. We have a really different time frames. Mm-hmm. So for instance, animals has like 7 times higher metabolic rates. Mm-hmm. So how does it really translate it that we are actually looking for the 10 minutes and 15 minutes? How does it equate to the human terms? And the human, of course, living up to the 100 these days, right? So, animal lives only 2 years. So this pathophysiology is fundamentally different. Mm-hmm. So even we find something, one molecules of the interest of a target is working in the animal. Do we really need to know what is the effective window for the human terms? And is this something that can be addressed in the multi-part?
That's a very difficult question, Sunky. and to be honest, I don't know the answer. I guess I'm at a stage now where I think that I don't know, and but I think there's so many other issues that we haven't addressed that it's difficult to understand whether the problems that we're seeing are because of the problem that you're describing, or because of all these other issues. So my approach, I guess, has been to try and address this low-hanging fruit of things that we know how to address and fix. And then, because essentially it comes down to the fact of should we be using animal models at all? And there are people who are very much against it. They say, then we've not translated, we shouldn't be using animals in research. And I'm like, I don't think we're at a stage now to say that we shouldn't be doing that, because I think there's lots of things that we can do to improve the rigour of preclinical studies. And if we do all these things and it still doesn't work, and then I think we then need to start working out. But that's definitely beyond my expertise in terms of the physiology and the pharmacokinetics and of the differences between animals and humans. I know that doesn't really answer your question, but it's because I don't know the answer.
Yeah, sure. I understand the difficulty of it, but hopefully that we could actually understand in the biological, pathological, in line between the preclinical and the clinical setting. Then maybe, this is one way to approach it. It's just my recommendation.
Yeah, no, and what you're saying makes sense, for sure. Thank you.
Hello, so my name is Esther. I'm a PhD student from the University of Utrecht, or UMC Utrecht. I was wondering, we just had a nice positive finding in our study as well, but it's like the first study we did. And then my question is, would you then recommend to first have it reproduced in different labs before starting multi-center trials, or would you say, go straight to the multi-center preclinical trial if you have a positive finding, because then you still have that external validity as well, and not everything is the same. Because I think you will also get the challenge where people are more against animal experiments, and if you do every step of the way, you're going to use a lot of animals. So what is your view? How would you see? Would you say first always reproduce and then go to multicenter, or would you say go for it and multicenter is better?
Yeah, I mean, I don't know. I think we waste a huge number of animals now with the way we're doing it. So I think what you're describing actually would probably result in fewer animals in the grand scheme of things. I mean, it's difficult just without all the information, because I don't know within your study how many of the different constructs that you looked at, you know, how many different outcome measures, the site, all that stuff. I would probably be more inclined to have some replication attempts first, just because of that kind of ramp up to, you know, the power that the number of animals that you then will probably require to do a multi-centre study. But it's not that black and white. I think you need to start thinking about the totality of the evidence, you know, thinking about things like the STAIR criteria and thinking about all the different types of, and it's unlikely that one lab would do that. And also there may be others who have done similar experiments that weren't positive and they never got published because of publication bias I was talking about that we're unaware of. I think that happens lots. So having that replication, I think, would address some of those questions but you know, you might have it might be so obvious that then you're like, well, why would we bother doing that? Let's just go to. So I don't think it's quite black and white.
It sounds logical. I was just wondering. And thank you very much.
No, thank you.
Hi, thank you. I'm Giuseppe Pignataro, Naples, Italy. We started a multi-center preclinical studies a couple of years ago on remote post-conditioning in stroke, and we registered it on preclinical UA and published it on BMJ Open. But the most difficult part was on the ethical committee approval. I don't know about the other countries around the world, but can you summarize this aspect? I think it's very important. We got a lot of problems with this approval in a multi-center.
Yeah, it's something that. So when we did multi-part, UK was still part of the EU. So we had, and it was an EU project. So we looked at the different ethical approval processes in different jurisdictions. And some countries had higher, I guess, requirements in the EU directive that then made it difficult. Some countries, just the concept of replication, you can't do an experiment that's being replicated, which I think goes against science, to be honest. I think it's that I can understand how it came about, but I think that it's counterintuitive when you really drill down into it. I didn't go into detail, but I did make the point to say ethical and regulatory approvals for multi-centre are very, very difficult to do. And I think part of our work needs to be, I guess it's almost kind of like lobbying and making the argument of why this approach is appropriate in the way that, you know, getting ethics for doing a multi-centre human study, the logic and the rationale is understood. And the reasons behind it in a preclinical thing are slightly different, but I don't think that rationale is understood. And we had, and I don't actually know what's the situation, because we had some Home Office, people who worked in the Home Office who are in charge of ethical approval in the UK, who are part of this EU project, who were part of EU activities, and I don't know if they're still involved kind of post-Brexit, who are really keen on supporting changes to support the ethical approval. But at the moment it is very difficult because of just, why you can't replicate. If one person's done it, that should be the answer. Like one experiment is truth and one experiment we know isn't truth.
We might leave it there. Thank you so much, Emily.
No, thank you. Thank you, everyone.
Our next speaker is Matthew Brook from NC3RS, and he's going to follow on from Emily really nicely and talk about the importance of guidelines in improving preclinical research. So thank you, Matthew.
Thank you very much. My name is Matthew Brook. I'm the Programme Manager for Reporting of Animal Research at the NC3Rs. As Renee just said, I'm going to follow up what Emily's very nicely introduced there, talking about some of the tools that are available to kind of help with preclinical research, including the ARRIVE guidelines, which will be my main focus, and the EDA, which Emily also mentioned. Before I get started, I thought it would be worth just taking a second to introduce the NC3Rs, the organisation I represent. The NC3Rs is the National Centre for the Replacement, Refinement and Reduction of Animals in Research. We are a UK-based scientific organisation. We're focused on the discovery and application of new technologies to replace, reduce and refine the use of animals in scientific research, which are the 3 Rs. We work with researchers and organisations across the life sciences sector, both in the UK and internationally. This includes universities, representatives from the pharmaceutical industry, chemical and consumer products industries, other research funders, other regulators, and so on. We are primarily funded by the UK government, and most of our budget goes into funding 3 Rs based research. However, in addition to that, we also have a number of what we call centre-led programmes, office-based programmes, led by our in-house teams. One of these is the experimental design and reporting team, of which I make a part, and which does some of the work that I'll be telling you about today. We have a lot of different resources based around the 3 Rs and kind of improving animal research. You can find them all at our website. I'll discuss them a little bit later as well. So, what am I going to talk to you about today? Since this is the education day, I've put these down as learning objectives. I want to talk about how research is reported at the moment, whether the way that research is reported is helpful to you if you're as a reader of a scientific publication or somebody that might want to carry out a systematic review, why it's important to report research transparently. Then I want to introduce you to the ARRIVE guidelines, which are a tool developed by the NC3Rs to help with preclinical animal research reporting. I want to talk about why it's important to report particular things that you can find within the ARRIVE guidelines. Then I want to take you through some of the tools we have available that will help with better reporting, and that will include resources focused on experimental design as well, such as the EDA. So, first of all, to understand why it's important to report preclinical research well, we need to have a little look at what some of the current issues with reproducibility in preclinical research are. Scientific publications reporting preclinical animal research very often lack important information, and this really limits how useful they are to readers and people, for example, looking to reproduce that work. Reporting research transparently and accurately is really a key contributor to reproducibility. As an example of this, I want to talk about a large project which we just wrapped up called the Reproducibility Project in Cancer Biology. This is a program that began back in 2013. It set out to replicate 50 high-impact papers in the field of cancer biology. Now, as I mentioned, this project is recently wrapped up and reported its results, which I'll talk about in a second. But what's interesting from my perspective is the way in which the project was forced to reduce its scope over time. As I mentioned, they started out trying to replicate 50 of these papers, and then sort of over the years they had to reduce the scope of this down first to 37, then to 29, and finally to 18 papers. The reasons for this were multifactorial. The main reasons for the reduction of this scope were, firstly, that materials were not available. There might not have been animals available or particular reagents and so on. But the more interesting reason for our talk today is that a lot of these papers they were trying to reproduce simply didn't contain enough methodological detail to allow the experiments to be reproduced. So, as I mentioned, this project recently wrapped up and published its results. In the end, they attempted to replicate 193 individual experiments, which came from 23 different papers. The major finding of their reproducibility studies was that effect sizes in their reproduction efforts were 87% smaller than the original papers. But again, what's interesting for me to talk about today was how easy it was for them to replicate the experiments that they tried to look at. So they coded each of these 193 experiments according to how easily they were able to replicate a complete experimental protocol based only on the information in the manuscript itself. As you can see, none of these 193 experiments, 0%, could be replicated without contacting the original authors for clarifications. And more than 50% of them required what they considered to be moderate, strong, or extreme clarifications. This really highlights why it's important to report accurately and in enough detail your experiments in the first place, particularly as when they went to get in contact with the original authors, a lot of authors, again, they coded how helpful they considered the authors to be. A lot of authors were very helpful, and they got on board with this project and helped out. 41% of authors that they considered to be very or extremely helpful. However, 32% of the authors they contacted either didn't respond, couldn't be contacted, or refused to help with these replication efforts. Really, the takeaway from this is that even this very highly impactful research can't be reproduced if it's not reported transparently and accurately. And this is particularly important as, in many cases, it might not be possible to get the clarifications you would need to reproduce an experiment. So, these known difficulties in reproducibility of research were a major driving factor behind the development of the ARRIVE guidelines. ARRIVE is an acronym. It stands for Animal Research Reporting of In Vivo Experiments. The ARRIVE guidelines are a tool that was developed by the NC3Rs initially back in 2010 to improve the reporting of biomedical research involving animals. The guidelines consist of a checklist of individual pieces of information, which we call guideline items, which are necessary to describe an animal study comprehensively and transparently. As I said, the original guidelines were published back in 2010, and then from around 2016, 2017 to 2020, the guidelines were comprehensively revised and updated with these revised guidelines, known as the ARRIVE Guidelines 2.0, released in July of 2020. There are many items in the guidelines, some of which I'll go into in more detail. The items cover things like internal validity. This includes items that ask for details of your study design, your sample size, how your animals were allocated to groups or treatments. The items covering reproducibility, for example, items asking for a comprehensive description of your experimental procedures, the animals that we use in your experiments, and so on. And some items ask for details of the scientific context and the relevance of the study. For example, details of the scientific background, the relevance in other species, and things like ethical approvals. The guidelines apply to all in vivo research, whether you're using mammalian species, fish, invertebrates, and so on. Because the purpose of the guidelines is to help transparent and accurate reporting, it doesn't matter whether or not you need ethical approval for the studies, for the guidelines to be relevant. So if you're using juveniles or you're using species that don't usually require ethical approval, that kind of doesn't matter. The guidelines are applicable to all kinds of in vivo study. Excuse me. So, since their original publication, again back in 2010, the guidelines have received strong support from across the scientific community. Over 1,000 journals, universities, organizations recommend or endorse the ARRIVE guidelines. This includes a lot of journals, quite prominent ones, such as the ones you can see here. It includes universities across the world. This is a little map showing the countries in which we have universities endorsing the ARRIVE guidelines. It also includes funders and learned societies around the world, some of which are quite highly influential. In the past, we have produced what we call little pocket-sized versions of the ARRIVE guidelines that you can keep on your desk or your lab bench, and we've sent those out for free. We've had requests for over 40,000 of those from over 50 different countries around the world. So you can see that the guidelines are quite popular. However, since their original publication, it was important for us to understand and look at whether or not they were actually having any impact on the quality of animal research reporting. Now, Emily mentioned a few minutes ago an example of a kind of study that looks at the quality of research reporting. And this is another example of one of these studies. This is a study that was published at the end of 2020. It used a tool called SciScore, which is an automated system for assessing published manuscripts for the presence of particular criteria. And what this study looked at was the presence of these criteria that are criteria associated with reductions in subjective bias in studies. This graph shows this graph is focused on animal research papers specifically, on 51,000 open access animal research papers published in 2018. And it shows the proportion of those papers that reported use of these specific criteria. You can see randomization in yellow, blinding in red, power in green. What you can see is that across all of these animal research papers, use of these criteria is still very low. So randomization, for example, of animals to treatment groups was mentioned in 37% of papers, blinding was mentioned in only 12%, and use of a power calculation to determine sample size was mentioned in only 7% of these papers. This graph on the right shows the results of all 1.6 million biomedical research papers. These are clinical papers as well as preclinical papers, so not just specifically animal research papers, over the period that they looked at them. So you can see the changes in some of these criteria across that time. So while you can see there is some improvement in these, for example, in randomization in yellow again, has improved quite substantially over that time. However, the level of reporting of these criteria remains still pretty low across all of these papers. So again, this kind of, this fed in to our revision, the NC3R's revision of the ARRIVE guidelines. So as I mentioned, by around 2016, 2017, we had a great deal of endorsement of the ARRIVE guidelines at the NC3Rs. Over 1,000 journals had endorsed them. However, there was not a great deal of evidence of actual changes in practice, in reporting practices. For example, there were a great deal of papers that were citing the ARRIVE guidelines paper, but were still not actually including any of the information requested by the ARRIVE guidelines in that paper. We wanted to understand the reasons behind this. So the NC3Rs in 2016 and 17 carried out a survey of in vivo researchers worldwide. There were two major findings of this survey. The first was that knowledge of the ARRIVE guidelines is associated with more rigorous study design and more rigorous reporting. This confirmed the findings of a previous study, which showed that knowing about the ARRIVE guidelines is associated with better study design, as evidenced by use of these markers for reducing subjective bias. Secondly, what the survey found was that poor reporting, i.e. missing information from papers, reflected the perceptions and the attitudes of researchers in many cases, rather than journal behaviour. What I mean by this is the reasons why a lot of things were not being reported were that, for example, researchers didn't really understand why they needed to be reported. They thought that it wasn't necessary to report some things, and they had perceptions like journals won't allow you enough space in your methods section to report all this important information, which in many cases is just not the case. So, based on this survey and other data, the NC3Rs convened a working group to revise the ARRIVE guidelines. This had 28 authors. It included representatives from funders, journal editors, statisticians, methodologists, researchers from academia and industry. As part of the revision process, the NC3Rs undertook an expert Delphi exercise in order to prioritise the items in the guidelines and work out which were the most important. This included 73 participants from 19 countries around the world. And once the guidelines were published as a preprint on bioRxiv, they were road tested by researchers in the process of preparing animal research manuscripts. Both the kind of breadth of this Delphi exercise and the road testing of the guidelines were there to ensure that the guidelines were really fit for purpose and were appropriate to their audience of researchers preparing in vivo studies. So, this resulted in the publication in 2020 of the ARRIVE Guidelines 2.0, the revised ARRIVE Guidelines. These are them here. So in comparison to the original ARRIVE guidelines, they're a little different. Some items were updated, some were condensed, a couple of new ones were added. But really the major change from the original ARRIVE guidelines in ARRIVE 2.0 was the prioritization of the guidelines into these two sets. On the left here, you can see the first of these sets, which we call the ARRIVE Essential 10. These are the items, the pieces of information that really are the basic minimum to include in any animal research manuscript. Because without these pieces of information, it's very difficult for you to assess the reliability of the findings presented in the study. They include things that I've mentioned already, like your study design, details of your sample size, randomization and blinding, procedures in animals, and so on. Alongside that, we have the second set, the recommended set. These are a set of items that complement the Essential 10. They add context to the study, and background, and so on. Reporting all of the items from both sets is considered best practice, but the purpose of prioritizing them in this way was to ensure that kind of initial focus by journals and researchers is on these most crucial items, the ones found in the Essential 10. In addition to this prioritization, and to address what I just talked about a few minutes ago, the kind of misunderstandings of some of the ARRIVE items, incorrect perceptions about them, the NC3Rs also published an extensive explanation and elaboration document alongside the revised guidelines. This contained background on each guideline item, the rationale for including it, why it's important to report that particular item, as well as examples of how to report each item well that already exist in the published literature. Excuse me. So, when can the guidelines be used? As I mentioned, the E&E, the explanation and elaboration document that was published alongside them, contains, in addition to information about each item, it contains quite a lot of useful information about experimental design and kind of how to plan and carry out your study. That means that consulting the ARRIVE guidelines can be very useful during the planning of your study, as well as when reporting it. In addition, it's useful to use the guidelines during the conduct of your study to make sure you're reporting all of the information you'll need to report it correctly, during the writing of your study, of course, and when the study is being reviewed. So, I wanted to just take a few minutes next to talk about why it's important to report some of the things in the Essential 10 and kind of the impacts that they have. So the first of these is the first item in the ARRIVE Essential 10. The first couple of items are asking you to describe your study design and your sample size. However, the sample size kind of by itself can often not be as useful as it could be if you don't describe what the experimental unit of your experiment is. So what do I mean by experimental unit? The experimental unit is classed as the biological entity subjected to an intervention independent of all other units. And your sample size, essentially your N number, are the number of experimental units you have in each group. It's sometimes not straightforward to identify what the experimental unit is in an experiment if it's not kind of explicitly stated. Sometimes an experimental unit can be a single individual animal, but sometimes it isn't. And I want to just look at a few examples of this to kind of describe kind of the difficulty of understanding what the experimental unit is. So as I mentioned, the experimental unit is the thing that's subject to an intervention independently of all other units. You may be carrying out, for example, a cell culture assay on the left here, in which you're providing your treatment, your intervention, as represented by this little syringe, to one well of your, in this case, 6 well cell culture plates. In this case, because the well is the entity that the intervention is being subjected to, your experimental unit is the well. You might take measurements on multiple cells from within that well, in this case, these 4 cells down here, but in this case, the experimental unit is the single well. In an animal experiment, as illustrated by this first mouse here, you might provide your intervention to a single mouse, and you might make your measurements, again, on that same single mouse, in which case the experimental unit is quite clearly the individual mouse. However, if your giving an intervention in this example, in this second mouse example, to a pregnant female mouse, and you're taking your measurements on the offspring of that mouse. Because you have provided your intervention or your drug treatment, for example, to the pregnant mouse, the experimental unit is the mouse to which you've given the treatment. And the same applies again in this fish example, this zebrafish example. If you're providing, for example, a drug treatment to an entire tank of fish, you might be taking measurements on individual fish from that tank, but because the treatment is being given to the entire tank, the tank is your experimental unit. It's fairly important to report what the experimental unit is clearly, because it will really make a difference to kind of how data is perceived. You would draw very different conclusions from a paper reporting an N of 10 versus an N of 1, even if that N of 10 really doesn't represent 10 individual experimental units. Yeah, and that's why it's important to report this experimental unit explicitly in your papers. Next, I want to talk about randomization. ARRIVE suggests that you report details of how, for example, your animals were allocated to groups, and in addition to that, how, if randomization was used, how randomization was achieved. So the important thing with randomization is that you need to use a validated method for it. Selecting, for example, animals in what we call a haphazard method. So imagining a cage of mice, if you just reach in and grab animals in whatever order you manage to get hold of them, that's what's called haphazard selection. That's really not truly random. The result of that is that you can kind of introduce systematic biases into your group, which I'll talk about again in a second. So really, to get around this, the important thing to do is to use a method for randomization that you can't have any influence over. This can be something really simple. You can, for example, use Excel to generate a list of random numbers. You can flip a coin. You can use, as in this really simple example, you can roll a dice. The most important thing is that you can't subconsciously influence the method by which you're randomizing these animals. Randomization is important for two key reasons. The first of these is that, as I just mentioned, it minimizes selection bias. So to kind of extend the example I was using a second ago, if you're just picking up your mice in whatever order you happen to be able to reach them, the result of that may be that you just end up with all of the slowest mice in one group in your experiment, which has introduced a selection bias to your experiment. The second of these is that randomization is a key assumption of your statistical analysis. Your statistical analysis is based on the idea that your groups are derived, animals or experimental units have been assigned to groups randomly from a uniform background population. And if you've introduced a selection bias to that, then you're going to end up with some difficulties with your statistical analysis. To really illustrate the way in which humans can kind of influence randomization, I just wanted to talk briefly about this little survey, which really illustrates how humans can't be relied upon to do random, as I put here. This graph shows the results of a really simple internet survey that was carried out way back in 2007 now on a blog called Cognitive Daily. In the survey, people were simply asked to pick a number between 1 and 20. And the results of the numbers the humans picked were compared to results from a computer using a truly random allocation. As you can see, if the numbers were being picked truly randomly, as there are 20 numbers, you'd have a 5% chance of each number being picked. And that's roughly what you can see with these white bars, which show the results from the computer's random selection. These oscillate roughly around this 5% line. However, when the humans were asked to pick a number, almost 20% of them picked number 17, and over 10% picked number 7. So there must be some kind of cultural reasons for this, why 17 is the most random number between 0 and 20. But what it really illustrates is that you cannot be relied upon to randomize by yourself. You need to use a method that is free from your own subjective biases. Next, I want to talk about the importance of blinding. So the ARRIVE guidelines recommend reporting whether who was aware of group allocation during each different stage of your experiment in your analysis. That is, was anyone blinded to group allocation at any point? To illustrate the importance of this, I wanted to talk about another study. This was a study carried out in Belgium on 157 veterinary medicine students. In this study, the students were trained to watch videos of pigs in their pens and to identify positive and negative social interactions between the pigs. Positive interactions with things like nose-to-nose contact with other pigs, playing and things like that, whereas negative interactions were things like biting of ears or other body parts or headbutting other pigs and so on. After this training, the students watched 5-minute video clips of pigs in their pens, and they counted the number of positive and negative social interactions that they saw. The students watched 2 different groups of pigs. There was a control group of pigs, and there were some pigs that were called high social breeding value pigs. These are pigs whose presence was known to increase weight gain in other pigs and were known to be kind of a positive influence on other pigs. You can see the results of this study on the right here. When the students watched the videos, they saw, as you might expect, the high social breeding value pigs in purple displayed more positive behaviours than control pigs. And in the bottom graph, you can see they also displayed fewer negative behaviours. But what was really interesting about this study is that, in fact, the video clips, the behaviour of the students watching the video clips was completely identical. The footage of the 2 groups of pigs had been visually manipulated. So, as in my little cartoon here, the footage of the high social breeding value pigs had been mirrored, the brightness had been changed, things like the dates and the numbers of the pens had been changed, to give the impression that these were 2 different sets of pigs, 2 different populations that were being studied. But in actual fact, the behaviour that was being shown on the 5-minute clips was completely identical. And the differences that were observed by the students between the 2 groups were entirely a result of their subjective perceptions. And this really underscores the importance of blinding, because you can get these statistically significant results entirely as a result of your own perceptions. And that's why blinding is important. So taken together, randomization and blinding are kind of the measures to reduce the subjective bias inherent in your manuscripts. You can really see the kind of cumulative effect of them from looking at the results of this study. This looked at the results of emergency medicine studies in animals and how these correlated with the use of randomization and blinding in the studies. You can see from the results here, studies that report neither randomization nor blinding are much more likely to report positive results. So this is in orange here, so a much lower proportion of negative results. Whereas studies that use both randomization and blinding have a much higher proportion of negative results, with use of one or the other giving an intermediate result. So you would expect that the proportion of, if all things being equal, the proportion of positive results in studies should be the same across all studies. So what this really suggests is that the use of randomization and blinding is avoiding the studies resulting in false positives. It's helping to eliminate the false positive results. Of course, this is kind of, these results, as Emily mentioned, kind of publication bias in favour of positive results is clearly another factor involved in this, but it is kind of quite stark, the impact that randomization and blinding has on false positive results being published. So, next I wanted to quickly talk about some of the resources that we've made available to accompany the ARRIVE guidelines that can kind of help with better reporting of animal studies. As I mentioned a couple of times, we have published this extensive explanation and elaboration document alongside the ARRIVE guidelines. And what we've done to try and make this kind of available to everybody, to make the importance of the ARRIVE guidelines easy to understand and access, is we've put all of the information from the E&E onto our ARRIVE Guidelines website in this kind of easily browsable format. So anyone that kind of goes to the ARRIVE website to look at the guidelines can see this this information kind of quite easily and kind of browse between the different items. This is rather than having it as a separate, very long manuscript where you'd have to go to access this information. You can find it all at our website, arriveguidelines.org. In addition, we've published a number of other resources that are designed to kind of help with use of the ARRIVE Guidelines. We have a webinar, which is kind of even longer than this talk and goes into even more detail about about the importance of reporting different items and so on. We have what we call author checklists. These are checklists that allow authors to indicate whereabouts in their manuscript you can find information related to each ARRIVE Guidelines item. You can submit these alongside manuscripts to journals. Some journals do request these, and we kind of encourage them to do so. Some journals have chosen to publish these alongside animal research manuscripts, so kind of this information is available. We have translations of the ARRIVE Guidelines. This is a little out of date. We actually have 2 more translations than this now. We also have Spanish and Greek available, in addition to these 6 you can see here. We have recently introduced what we're calling a compliance questionnaire. This is designed for journals and it consists of operationalized versions of the ARRIVE Essential 10 that allow journals or other interested parties to assess compliance with the ARRIVE Essential 10 in a kind of quick and streamlined fashion. And we also have, again, recently introduced something that we're calling ARRIVE Action Plans, which are lists of kind of useful activities that different stakeholders can undertake to kind of promote ARRIVE in their institutions. So we have action plans for journals, universities and funders. So if you represent one of those and would like to know more about what you can do to help promote the ARRIVE Guidelines, please let me know. Then lastly, I want to talk about something that Emily mentioned a little bit earlier on, some of the resources we have available for helping to design your animal experiments, including the EDA. So briefly, we have a wide range of resources dedicated to both the 3Rs and specifically to experimental design and reporting on our newly redesigned NC3Rs website. You can filter through all our resources. We've got hundreds available. They include useful, as well as information about reporting. They include, we have resources covering other useful information like carrying out systematic reviews, carrying out pilot studies, which I've misspelled there, apologies, sample size calculations and statistics, among other things. But what I just wanted to spend a couple of minutes talking about at the end now is our experimental design assistant, the EDA, which Emily mentioned earlier. The EDA is an online tool for researchers, which is designed to help you design better in vivo experiments. It was designed by experts in experimental design, statisticians, and in online web programming. And it's really designed to help increase the use of rigorous study designs and help you to design better studies yourself. You can find it at eda.nc3rs.org.uk or just by searching for the EDA. So, what does the EDA do and how does it work? So we like to say that the EDA is like having a personal statistical assistant to help you design your experiment. The program uses computer-based logical reasoning to help you design your in vivo experiment better. It will give you advice on the most appropriate statistical tests for your experiment. It will help you to determine the sample size you should use in your experiment by helping you to carry out a power calculation. It provides dedicated support for randomization and for blinding as part of your studies. For example, it will generate a randomization sequence and it will send it to somebody other than you who is helping you to carry out your randomization. So it can help you with randomization and it can help blind you to the randomization. And it will also produce a summary report that will kind of give you feedback on your experimental design and ways in which it could be improved. So this down here in the bottom right is an example of an EDA diagram. And I thought it would be worth just having a quick look at how the EDA works and the kind of thing that you will get out of it if you decide to use it. So in the EDA, all experiments can be represented as one of these diagrams. So in the diagram, these individual shapes, or we call them nodes, are used to build up the diagram. And you can use these kind of like Lego blocks. You can arrange them in all sorts of different ways. And in that way, you can build up really a diagrammatic representation of any kind of experimental design. Each individual node contains kind of detailed information about the different steps of an experiment, and the arrows between the nodes kind of show how they link together. I'll just take you through briefly this example we've got here. This shows a very, hopefully very straightforward two-group comparison experiment in which animals are subject to interventions and then have their blood glucose measured. So if we read, this mouse is a little insensitive, but if we read from the left here, we start off with our pool of animals. You can see that that's made up of experimental units that are individual mice. These animals are subject to randomization in this circular node into two groups, Group 1 and Group 2. In these purple nodes, the groups are subject to interventions. In this case, these are pharmacological interventions. Group 1 has a vehicle control injection, for example, whereas Group 2 is subject to a drug treatment. After this, both groups have their plasma glucose measured, and then again, both groups, the outcome measure for both groups is the level of glucose. This feeds into the analysis, and yet the EDA will then recommend an appropriate statistical test for you to use for this experiment. This is quite a straightforward experiment, obviously, but the EDA can accommodate some really much more complicated study designs and is really a useful tool for you to design better experiments. As I mentioned before, the EDA uses kind of computer-based logical reasoning, so it can once you've kind of designed your experiment, it can give you feedback on it, on kind of ways in which it can be improved and appropriate tests and so on. So I just want to talk very briefly right at the end on how these kind of couple of resources fit together. So experimental design and reporting kind of superficially on the surface of it address two different stages of the animal research process. So experimental design, you would think, would come at the start, you would design your experiments, and then reporting would come at the end. However, the focuses, kind of experimental design and reporting are inextricably linked together, and the tools that the NC3Rs have made available, i.e. the ARRIVE guidelines and the EDA, are quite clearly linked to each other. As I mentioned, the ARRIVE guidelines provide extensive recommendations on good experimental design, and the EDA, use of the EDA, is a really good way to fulfill a lot of the reporting recommendations laid out by the ARRIVE guidelines. I should have mentioned a second ago that you can export your EDA diagram at the end, and you can actually use that as a way of communicating your experimental design, both kind of between researchers and with journals, if you want to. And we're looking at actually improving the way in which the EDA diagram can be exported to make it easier to actually include EDA diagrams in published manuscripts. And that is because kind of the EDA diagram is so much more explicit than a text description of an experiment can be, and it's a really kind of clear diagrammatic way of showing your experimental design. So that's all I'm going to talk to you about today. If you'd like any more information, please feel free to get in touch with me, either at my personal address or we have an ARRIVE email address. Please feel free to visit our websites, nc3rs.org.uk or arriveguidelines.org. So thank you very much.
Can see that the guidelines have made quite a tour around the world there.
They have, yeah. They've been all over the place. I've got some with me today. I meant to hold them up when I talked about them. But
So thank you, Matthew. I'll open for questions now.
Hi, Nestor. Yes, I have a question and kind of a philosophical consideration. The question is, you've shown that over 1,000 journals, for example, have endorsed the guidelines, but I wonder how many of them have enforced them.
Well,
very little, because I guess in other of your slides, you have shown that for many of your indicators, most of them, if not all of them, didn't reach even 50%. So the question is, why the journals that are actually, you could say that the bottleneck, right? Because if 100% of the journals were enforcing this, every researcher will be forced to follow them, because they will not be able to publish, right? But that doesn't happen. Why the journals don't enforce it? Why do you think it's not in their interest to do it? And that triggers, if you allow me to finish, that triggers a little bit the philosophical question, and is whether imperfect science is not good. I'm not talking about bad science, I'm talking about imperfect science. And one clear example is, for example, blinding. You are in a lab where you have one postdoc preparing the drug, injecting it in the mice, and doing the imaging. Blinding is just not possible, because you don't have 3 technicians to do it. That doesn't mean that the research is bad. It's not optimal, it's imperfect if you want, but it's not bad. And science does not evolve only with optimal and super nice design experiments. It evolves also with imperfect experiments. That's kind of the philosophical consideration.
Okay, so there's a couple of questions there. So I guess to kind of address your last point, and this is probably something I should have mentioned in a bit more detail. There are, in many cases, good technical or logistical reasons why you can't do some of the things recommended by ARRIVE, why you might not be able to randomize, or you might not be able to blind, for many reasons, a lot of which are perfectly valid. I should really sort of emphasize that in order to kind of comply with the ARRIVE guidelines, you don't have to have blinded, you don't have to have randomized. You just need to report whether or not randomization and blinding, for example, took place. You don't need to have done a sample size calculation, but you should kind of report how you chose the number of animals you used. That could be that you had 3 mice that available. So there's the question of like good versus bad versus imperfect science is kind of slightly separate from the question of good reporting, which is what the ARRIVE guidelines are primarily focused on. Because kind of good reporting, kind of whether and how these things took place, is important so a reader can kind of assess how reliable they find the study. They can assess kind of how they can assess the quality of the study by whatever metric they want to assess it by. To your kind of circle back to your first point, enforcement of the guidelines and getting people to get on board with actually using them is kind of, to an extent, my full-time job now, is trying to get people to use the guidelines and figuring out different ways of doing that. So from our perspective, there's a lot of things we're kind of trying to do. So we're trying to make it easier to assess the guidelines. So that's what we've done with things like our compliance questionnaire, and we're looking at kind of technological solutions as well for automated assessment of guideline compliance that would make it a lot easier for journals to do it. Why journals don't do it? There's a multitude of factors. So, there are, for example, financial resources, human resources factors involved. Some journals are, of course, big operations with large editorial staffs. Some journals are run part-time by academics. So there's not a kind of one-size-fits-all answer to that question, but it is something that we are kind of actively working on and trying to kind of implement ARRIVE at all these kind of different levels, at the level of the researcher, at the level of kind of getting universities involved in it and other research institutions, getting journals involved. Kind of funders as well can do useful things with this. A lot of funders mandate use of ARRIVE, but again, there's no kind of follow-up to that. There's a lot we can do. There's a lot we are trying to do. But as for the kind of underlying reasons, I think they're quite myriad, if I can say that. I hope that answers your question.
So, Michael, just one final question before we wrap up for the break.
Yeah, I'm just kind of wondering, this morning we heard about multipart, which is essentially an attempt to impose clinical trials discipline onto preclinical studies. And I'm just wondering how the ARRIVE guidelines were arrived at, because if I was trying to do it, I would start with ICH GCP, and I would start with the enormous amount of guidance from the regulatory agencies about how to conduct adequate, well-controlled studies, rather than taking a group of academics from the preclinical stream and reinventing the wheel. It just seems inefficient to have one silo deciding about what is the best way to do preclinical research if the intent is to translate to clinical, and the wealth of knowledge and the wealth of guidance already exists in the clinical world.
Yeah, so as to kind of how the guidelines were developed, I should say that this kind of wasn't an activity entirely carried out by academics. So we had a lot. The working group for the Arrive revision, for example, contained a lot of journal editors. It contained funders' representatives to try and come up with something that worked for all of these different groups rather than just academics. And I guess I should kind of, there is a bit of a distinction between kind of the performance of research, carrying out research in the right way and reporting it in the right way, which is really what the Arrive guidelines are focused on. So, so that, I mean, that's the major focus of Arrive, is kind of getting the information into the manuscripts, and the kind of good experimental design information comes along with that, because it's important for researchers to understand why you should be reporting these things and why these things are important in order for them to understand why they should report them. So yeah, I'm not sure if that answers your question at all, but I kind of can't speak to exactly the mindset of the researchers developing the guidelines at the time and the working group, but I would hope that those considerations came into play.
Okay, with that, we might close the session for now. If you have any additional questions, please come and speak to our speakers during the break, or if we can have a few minutes at the end, we can revisit that as well. So for now, thank you, Matthew and Emily, and we have a half an hour break in the exhibition hall now. So thanks, everyone.